Once again, we begin with the structure of the cell. Are you able to see the highlighter? Okay, now it is having a red color. This is the structure of an animal cell. You can see the nucleus and the cytoplasm and all the organelles. For oxidation, the fatty acids are to be transported from the cytoplasm into the mitochondria. You see here mitochondria is meant for various oxidative activities, and along with that one of it is fatty acid oxidation. And we found earlier that this fatty acid is activated to fatty acyl coenzyme A before being transported into the mitochondria. And we will see once this fatty acid reaches the mitochondrion, how is it oxidized? Beta oxidation of fatty acid. Why do we call it beta oxidation? It's very simple. And why is it called oxidation? We call it oxidation because it involves removal of hydrogen. Removal of hydrogen is oxidation, while addition of hydrogen is reduction. Here, the hydrogen atoms being removed from these fatty acids are carried away by means of hydrogen carriers. Here you can see FAD becomes FADH2. FAD is removing two atoms from this substrate and it becomes FADH2. Similarly, you can see here in the third step, NAD plus is removing two hydrogen atoms to become NADH plus H plus, and that hydrogen atoms are removed from this substrate. So because of the removal of hydrogen atoms, this is oxidation. This pathway is oxidation. And why do we call it beta oxidation? Because you see here, this is the fatty acyl coenzyme A. Here, this carbon is alpha carbon. This first carbon is the carbonyl carbon. Don't count it. This is the alpha carbon. This red colored one is the beta carbon. These oxidation reactions are taking place on the beta carbon. That's why we call it beta oxidation. So beta oxidation of fatty acids. How many reactions are there? 1, 2, 3, 4 reactions are there. At the end of the fourth reaction, a new fatty acyl coenzyme A is formed, which contains two carbon atoms less than the first fatty acid which was present at the beginning. So here reaction number 1, 2, 3 and 4. In the fourth reaction, a molecule of acetyl coenzyme A is released. It contains two carbon atoms. These two carbon atoms are removed from this fatty acyl coenzyme A. That means this newly formed fatty acyl coenzyme A at the end of the fourth reaction has two carbon atoms shorter. You can see it very clear. Two carbon atoms shorter. Let us see the reaction number 1. Look here. Formation of trans alpha beta double bond through dehydrogenation by the flavoenzyme acyl-CoA dehydrogenase. So an enzyme called acyl-CoA dehydrogenase. Actually, what's that reaction being catalyzed? It is very clear. It's a dehydrogenation reaction. From fatty acyl coenzyme A, two hydrogen atoms are removed. Those hydrogen atoms are removed and carried away by FADH2. And which are those hydrogen atoms? Look here, it's very clear. Here CH2 was there. Here also CH2 was there. How many are here? Only one hydrogen is here. Only one hydrogen is here. That means one of this red hydrogen and one of this black hydrogen is removed by this FAD and it became FADH2. So between those two carbon atoms, a double bond is now formed. What do we call it? Trans delta 2 enoyl coenzyme A. Delta 2 enoyl coenzyme A. Delta 2 indicates the position of the double bond. And reaction number 2, it's a hydration reaction. Hydration means addition of water. It is across this double bond. The enzyme is always hydratase, enoyl CoA hydratase. What is produced? You know, water means H2O. H2O means H and OH. Here, H is there, OH is there. And where are they added? Earlier here only a CH was there. Now here we have a CHOH. So the OH of water is added here and the H of the water is added here. So CH became CH2 because a hydrogen is added there. And this CH became CHOH because this OH of the water is added here. Now better we call it beta hydroxyacyl CoA. In this textbook it is shown here as 3L hydroxyacyl CoA. You may also call it beta hydroxy acyl coenzyme A. What's the enzyme? It is enoyl CoA hydratase. And reaction number 3. Here again a reaction of oxidation is taking place. And this is another dehydrogenation reaction. Hydroxyacyl CoA dehydrogenase is the enzyme. Here also two hydrogen atoms are removed. They are carried away by NAD plus. It becomes NADH plus H plus. So from where are the two hydrogens removed? One hydrogen from here, one hydrogen from here. So two hydrogen atoms are removed from the beta carbon atom. That's why now only an oxygen atom is remaining here, and it is bonded by a double bond. So it is a beta keto compound. What is formed is beta keto acyl coenzyme A. And what's the name of the enzyme? It is hydroxyacyl CoA dehydrogenase. And in the fourth reaction, now here we have a keto compound. Here we have a 2 carbon tip is there. This is removed in the form of acetyl coenzyme A. Look here, this is CH3COS CoA. Here we have a CH2COS coenzyme A. Remember, this is actually a double bond. This is not a single bond, but this is a double bond. Look here, this was a double bond, this was a double bond, this was a double bond. So this also is a double bond. The enzyme is beta keto acyl CoA thiolase. Thiolase means coenzyme A is added here. Coenzyme A is added here, and there is a breakage here. There is a breakage, and here at this point the coenzyme A is added. Now we have a new fatty acyl CoA, and this CH3COS coenzyme A is removed here as acetyl coenzyme A. Now I have a question. Here only CH2 is there. Here we have CH3. From where came the extra hydrogen? Remember, there is a hydrogen atom here. Don't forget it. So this is a thiolase reaction, and acetyl coenzyme A is removed. These two carbon atoms were earlier part of this fatty acid. Now the newly formed fatty acid has 2 carbon atoms less. Actually here they give n number of CH2 groups. Here all they give n number. So this is not a correct depiction. They should have given here n minus 2. CH2 n minus 2, because 2 carbon atoms are removed here in the form of acetyl coenzyme A. And this fatty acyl CoA again undergoes the same reaction. Reaction number 1, 2, 3, 4. Again a new fatty acyl CoA is formed, which contains two carbon atoms less. Again the new fatty acyl CoA undergoes this reaction. This reaction is continuing. Step by step the reaction continues. Finally, the remaining fatty acyl coenzyme A is broken into 2 molecules of acetyl coenzyme A.